The science of genetics began in a monastery garden in Brno. Between 1856 and 1863, Gregor Mendel cultivated and crossed nearly 30,000 pea plants, tracking 7 traits across generations. He found that inheritance followed precise mathematical ratios. His paper, published in 1866, described the laws of heredity. It was ignored for 35 years. Mendel's work was rediscovered in 1900, simultaneously and independently by 3 botanists who found their own results matched his. The field of genetics was born overnight. Within a decade, chromosomes had been identified as the physical basis of heredity. The gene was still a concept, the molecule had not been found. The molecule was identified in 1944 by Oswald Avery. DNA carried genetic information. The structure was revealed in 1953. James Watson and Francis Crick at Cambridge built a molecular model using data from multiple sources, including crucially the X-ray crystallography work of Rosalind Franklin and Maurice Wilkins at King's College London. Franklin's Photo 51, an X-ray diffraction image of DNA, showed the helical structure with extraordinary clarity. Watson saw the photo without Franklin's knowledge or permission, provided by her colleague Wilkins. It was instrumental in his and Crick's model. Franklin died of ovarian cancer in 1958, 4 years before Watson, Crick, and Wilkins received the Nobel Prize she could not share. Watson and Crick's Nature paper of April 25, 1953 is one of the most important documents in the history of science. It described the double helix, 2 complementary strands of nucleotides wound around each other. It ended with one of the greatest understatements in scientific writing. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. The structure explained the mechanism. Each strand of the double helix serves as a template for a new strand. When a cell divides, the helix unzips and each half is copied exactly. The genetic code is preserved with extraordinary fidelity across billions of cell divisions. Mutations are errors in this copying process, and mutations drive both evolution and cancer. Reading the genetic code required new tools. Frederick Sanger developed chain termination sequencing in 1977, a method to read the sequence of DNA bases. He had already won a Nobel for protein sequencing. DNA sequencing earned him a second, in 1980, one of only 4 people to win 2 Nobel Prizes. Sanger sequencing made the Human Genome Project possible. The Human Genome Project launched in 1990, a 13-year international effort to sequence all 3 billion base pairs of human DNA. It involved 20 research institutions across 6 countries. At its completion in 2003, it had produced the reference sequence of the human genome and established the infrastructure for the genomic age of medicine. The genome sequence transformed medicine's understanding of disease. Thousands of genetic variants associated with cancer, heart disease, diabetes, and psychiatric conditions were identified. Pharmacogenomics emerged, the understanding that genetic variation explains why the same drug works in some patients and fails in others. Medicine could finally begin to address individual variation rather than treating all patients identically. The ability to edit the genome came in 2012. Jennifer Doudna and Emmanuelle Charpentier adapted a bacterial immune mechanism, CRISPR-Cas9, into a precise gene editing tool. For the first time, specific sequences of DNA could be targeted and altered in living cells. Doudna and Charpentier received the Nobel Prize in Chemistry in 2020. The therapeutic applications are only beginning. Personalized medicine has transformed oncology. Tumor genome sequencing identifies the specific mutations driving a patient's cancer. Targeted drugs, designed to block those specific mutations, replace blunt chemotherapy. A lung cancer patient whose tumor has an EGFR mutation responds to erlotinib. The same patient without that mutation does not. The genome tells the oncologist which tool to use. Gene therapy has moved from promise to reality. Treatments for spinal muscular atrophy, sickle cell disease, and certain forms of blindness now use viral vectors to deliver corrected genes directly into patient cells. The first patients treated at birth for conditions that previously meant early death are growing up healthy. Mendel's pea experiments have become medicine for individual patients. From Mendel counting peas in a monastery garden, to Watson and Crick's double helix, to Sanger reading the sequence, to the Human Genome Project mapping 3 billion bases, to CRISPR editing them precisely, to gene therapy treating patients at the molecular level. 150 years from inheritance patterns to rewriting the code of life. I am Takku. We began healing through observation, we now rewrite the very code of life. Subscribe to Wisdom of Takku, because every era builds upon the wonder of the last.